Today, I'm happy to share our recent findings on bovine mycoplasmosis. At first, I would like to introduce this disease. Bovine mycoplasmosis is caused by Mycoplasma species, especially M. bovis. The symptoms of M. bovis infected cattle are chronic pneumonia, otitis, mastitis, and arthritis. These animals show poor growth rate and decreased milk production. Unfortunately, any vaccine cannot prevent this disease effectively, and antibiotic for treatment is limited due to drug resistance. So now we need new approach to control this disease. Our interest is immunosuppression. Several reports show M. bovis have an immunosuppressive character in vitro, such as, for example, suppressing lymphocyte cell proliferations and cytokine production from monocyte. This characteristic could be associated with disease progression. However, we still don't know the detailed mechanism of immunosuppression. So, we focused it on PD-1, PD-L1, immunoinhibitor molecules. These molecules are regarded as a common mechanism of immunosuppression in several chronic infections. PD-1 is expressed on effector T cells, and its ligand PD-L1 is expressed on the infected cells. When PD-1 binds PD-L1, negative signal from PD-1 inhibit T cell effector function. This suppression is called exhaustion. Exhausted T cell cannot reject pathogen. But fortunately, this interaction is reversible. So, by blocking this interaction using specific antibody, we can reactivate T cell. And reactivate T cell restore their effector function, and they can remove the pathogens. In this study, in order to elucidate the mechanism of immunosuppression, we investigated the role of PD-1, PD-L1 pathway in M. bovis infected cattle. Our study was divided in two parts. First part is analysis of in vitro infection models, and second part is analysis of clinical samples from naturally infected cattle. At first, I would like to show the result of in vitro infection models. In this analysis, firstly, we collected the PBMCs from healthy cattle and cultured with live M. bovis. After that, we investigated the PD-L1 expressions and its inducer. This slide shows the result of PD-L1 expression analysis by flow cytometry. As you can see here, PD-L1 expression on monocyte treated with M. bovis was higher than untreated monocyte. Yes. In other words, M. bovis induced the upregulation of PD-L1 expression on monocytes. Next, to elucidate the mechanism of PD-L1 expression, we focused it on prostaglandin E2. PGE2 is an important lipid mediator in inflammations and immunosuppression. Recent report showed PGE2 is one of the inducer of PD-L1 expression. So next, we investigated the PGE2 production in our in vitro model. We cultured PBM sheets from healthy cattle with live embovis and measured PGE2 production in culture supernatant. As a result, PGE2 production, we found that PGE2 production was increased by embovis. Interestingly, this PGE2 production was positively correlated with PD-L1 expression on monocytes. So it suggests embovis could upregulate PD-L1 expression by PGE2. As a second part, we analyzed the clinical samples from naturally infected cattle. Embovis infected cattle were diagnosed at Animal Medical Center of Lucknow Gakuen University. The symptoms of these cattle were pneumonia, otitis, and arthritis. First of all, we collected the PBM sheets and evaluated the T-cell response. After that, we conducted the expression analysis of PD-1, PD-L1, and blockade assay of PD-1, PD-L1 pathway. Okay. In order to evaluate the T-cell response, we cultured PBM sheets from infected cattle with T-cell stimulator and measured interferon gamma production in culture supernatant. As you can see here, interferon gamma production was reduced in embovis infected cattle. Interferon gamma is an important factor to protect the bovine mycoplasmosis, so it suggests T-cell response against embovis might be decreased during embovis infection. Next, we investigated the PD-1 expression on CD4 positive T cells and CD8 positive T cells. Compared to uninfected cattle, the proportion of PD-1 positive T cell was significantly increased in embovis infected cattle. Interestingly, the proportion of PD-1 positive T cells was negatively correlated with interferon gamma production. So it suggests PD-1, PD-L1 pathway could be involved in the modulation of T-cell response in embovis infected cattle. And next, we analyzed the PD-L1 expression on monocyte in embovis infected cattle by flow cytometry. As you can see here, the proportion of PD-L1 positive monocyte was increased in embovis infected cattle. Finally, we conducted the blockade assay of PD-1, PD-L1 pathway. We cultured PBM sheets from infected cattle with anti-PD-1 or PD-L1 antibody and stimulated with heat-killed embovis. After that, we measured the interferon gamma production in culture supernatant. Okay. As a result, we found a blockade of the PD-1, PD-L1 pathway deactivated embovis specific interferon gamma production. Okay, let me summarize our result of study. During embovis infection, the proportion of PD-1 positive T cells and PD-L1 positive monocytes were increased. Then, T cell exhaustion was induced. As a result of this, interferon gamma production was decreased and immunosuppression occurred. On the other hand, by blocking the PD-1, PD-L1 pathway using specific antibody, we reactivated interferon gamma and bovis-specific interferon gamma response. So, in conclusion, the PD-1 PD-L1 pathway could be a novel therapeutic target for bovine mycoplasmosis. Okay, as a future work, in order to elucidate the detailed mechanism of immunosuppression, we will conduct the further analysis of PGE2. In addition to this, we are planning the clinical trial of therapeutic antibody using M-bovis-infected cattle. We believe our study contributed to the control of this disease. Thank you for your kind attention.